So hello everyone. So welcome to our webinar. We will wait for a few minutes for our other participants to join. And then till then I will start the introduction. So Christian Biosystem, established in 2003, is a biotech firm based in Mumbai, focuses on understanding our customers' project from a technical standpoint to provide cutting-edge solutions to scientists to support their research. We have a strong background and expertise in immunology and working with biopharma, CROs, vaccines, and academia at pan-India level. I'm Dr. Swati Chitrangi, working in life sciences field, both in academia and industry for over 16 years, focusing on drug discovery, tox sciences, and product management. My core expertise is to bringing the in vitro solutions in market, particularly humanized models and regulatory compliant assay platforms that support pharmaceuticals, biotech, and academic research. By training, I hold a PhD in biological sciences along with executive business training from IIM Lucknow. Over the years, my work has spanned 3D cell culture systems, organoid, in vitro toxicology, and translational tox projects. I have opportunity to lead teams and projects at Advances Group, Yash Raj Biotechnology, IIT Bombay, and also I carry out internationally recognized research in IIST Japan. I also got the opportunity to share insights at various industrial platforms like CPHI, IPAC, ISSCR, AIMS Delhi, and I am a strong advocate for non-animal human relevant testing platforms in line with global regulatory frameworks like FDA Modernization Act. I've also published a lot of peer reviews, journals. So I will introduce now the Epithelix, which is a Switzerland-based company. So Epithelix is a Geneva-based biotech company specializing in non-animal in vitro respiratory models for drug development and toxicity testing, a European leader in reconstituted human respiratory tissue models. Epithelix developed the proprietary muciliaire technology, enabling long-term physiologically relevant tissue models that closely mimic native human airways. Epithelix supports pharma, chemical, and medical sectors and offers CRO testing services globally. We have Dr. Samuel Constant with us, who is the co-founder and CEO at Epithelix with us today to give us insight on advanced immunocompetent primary human airway models for respiratory disease modeling. So Dr. Samuel is co-founder and CEO of Epithelix, a synthetic organic chemist. By training, Dr. Constant has over 20 years of international research experience, having worked across Switzerland, France, and United Kingdom and Japan. He has extensive expertise in vitro test method development, particularly in advancement of human relevant respiratory models for biomedical research and drug development. Dr. Constant has co-authored more than 150 scientific publications and has received 18 awards recognizing his contribution to scientific research, technological innovation, and business development. His work continues to support the development of advanced in vitro models that improve translational research and reduce reliance on animal testing. I request everyone to be on mute and you can drop your questions in the chat box. We will address the same during our QA session. And now I hand over this session to Dr. Samuel to continue with the presentation. Thank you, Dr. Samuel, for your insightful presentation. And I'm looking forward to it.
Yes, thank you very much for your very kind and extensive introduction. So I have to say that I'm an honor and really pleased to share with you some of our advance at Epithelix. So first of all, do you see my screen?
Yes, sir.
Yes,
it's okay. It's okay. Perfect. So, what I will try to summarize in this next for 40 minutes is some kind of journey that we have achieved at Epithelix already, 20 years journey, where we are at Epithelix provider of respiratory tissues and in vitro solutions, and our aim is to provide to the worldwide relevant, innovative, and also predictive model which could be applied everywhere as decision-making tools. And what I'm going to present you is the latest advance in terms of immunocompetent primary human airway models for respiratory disease modeling. So at Epithelix, we are working only on modeling respiratory diseases. Why? Because clearly respiratory pathologies are constantly increasing. So we can name a few, so this is not an extensive list, but still the major ones are chronic obstructive pulmonary diseases, COPD. According to the WHO, it's estimated to around 280 million of persons. And is projected to be the third cause of death in the 2030s. Another devastating respiratory disease is obviously lung cancer, where there is approximately 1.8 million deaths per year. Asthma has also a strong impact in their life and also in cost for the whole economy, global economy, and it's estimated to around 400 millions of people worldwide. Respiratory allergy is also really increasing. Just give you a focus highlight on a specific country, which is France, for example. Just imagine that in the 1960s, there was approximately 3.8% of the population suffering from respiratory allergy. And now, in the latest report of 2021, it's estimated to be 30%, meaning that there's a 10 times more respiratory allergy. So it needs to be also strongly considered. And obviously, there are also a lot of viral and also bacterial infections also which are also clearly affecting people. I'm sorry because I hear a noise. Maybe someone could please could you shut down your mic, please? If
Anitesh,
you know, yeah, thank you.
Ravi,
Okay. So I was talking about SARS-CoV. Yeah, thank you. SARS is also we all know the main global problem due to this infection, which touched 775 millions of people and resulted in 7 million deaths. So we are interested here in recapitulating part of the respiratory tract in vitro, and just want to discuss a bit about the anatomy. The respiratory model, so human respiratory model, is mainly based on three types of epithelium, which are the first cellular contact point when we breathe something. So pretty clearly, in the upper respiratory tract, the nose, trachea, and bronchi, you have one type of epithelium, which is here based mainly on basal cells, ciliated cells, also goblet cells, and their presence of cilia on top and mucus covering this to ensure that we have efficient mucociliary transport. And now, if you go deeper into the respiratory tract from generation 3 in the bronchi, you end up with an epithelium which is a bit different. It's thinner and it's composed including another type of cell, which is cuboidal club cells on top of the one that I presented before. And finally, in the distal area, where in the alveoli, where the gas exchange occurs here, the epithelium is completely different. You have mainly two types of cells composing the alveolar sacs here, the 18-1 and 82, pneumocyte of type 1 and pneumocyte of type 2. And it's fair to say that here you have immune cells which are also present to ensure clearance here in these regions, which are the alveolar macrophages. While in the upper respiratory tract, the clearance mechanism is mainly driven by the presence of cilia, which are active. predictive. So clearly, this ciliation is really important when you are to model so the respiratory diseases. So what we have done at Epithelix is the following. So within the past 20 years, we have been modeling the nasal mucosa, the trachea, and also the bronchi, with a system which has a trade name of mucilaire. So mucilaire is made of the three type of cell: basal cell, ciliated cell, goblet cell, cilia, and mucus on tops. Is made of primary human cells. Okay, in the small area region, so we have developed also a model which is called small air, which bears the same type of cell as I mentioned previously, but also including cuboidal club cells. Finally, we have been successful in the development of a model for computing the alveolar compartments. So it's called alveolar, which is made of pneumocyte of type 1, type 2, co-culture with endothelial cells, and which is producing surfactant here. So how do we prepare this model? So all of these models are prepared in laboratories, and we start with primary human cells, okay, taken from surgical operations. So for the nasal cells, we use nasal polyps, and we undergo cell selections to select the progenitor cells that we could cryopreserve. So we have a bank of cells here, a bank of donors at Epithelix, but we could use these cells and seed them into macroporous filters, so which are here a classical transwell system, and we switch the cells to the air-liquid interface, and they progressively differentiate into a pseudo-stratified epithelium, so which bear all the cell types that I mentioned before. So the epithelium is tight, and you have clear cilia on top, and also presence of mucus on top. Okay. Um, no, I'm sorry, I can hear a noise. Maybe, yeah. Uh, may I ask you, please? Thank you. Thank you. Okay, for now concerning tracheal and bronchial cells, so we start now with wall lung, and we can undergo the same type of process to reconstitute now a tracheal versions of the mucilaire, and we have also a bronchial version of mucilaire. So all of these donors, all of these models could be made from single donor, but we have also developed an interesting aspect where we could pool the cells from different donor at the same time. So here, what we call nasal pool, where we mix cells from here 14 different donors to minimize the potential inter-individual variability from these tissues. And obviously, you could also take advantage of the genetic background of the patient, or also pathologies, where we could generate also disease versions such as cystic fibrosis, allergic rhinitis, asthma, or COPD. So all of these upper respiratory tract models, so the mucilaire, so you have cilia really everywhere, but the cilia are functional, and you can record their activity using, for example, software that we have developed and we are commercializing also, which is called Cilia X, that we can here record some bitmap, eating bitmap, and then you can derive some frequency distribution. And what you can gain here are some data here, reading here the cilia beating frequency and also the percentage active area. So how could we use this? So clearly, if you shift the cilia beating frequency to the left, this could in turn have a consequence that the xenobiotic could stay longer. So it could be detrimental in a way. So that means that it could be a tox endpoint if you shift this frequency to the left. Okay, so but cilia beating is one part of the problem in the mucociliary clearance. So mucociliary clearance could be done here on an other and we has another assay where we could have some micro beads on top of the mucus, and they could move thanks to the impulsion of the cilia beating, and you can derive here a velocity distribution which is here, and which is interestingly the value are really close to what are reported in human bronchoscopy. So basically, if you use wall type or non-disease cells, or so you could do this experiment. You have this transport, which is in the normal range, as we have shown before. But now, if you reconstitute epithelia based on cystic fibrosis, so cystic fibrosis is the first genetic background in terms of predominance, but so it has a consequence due to the let's say, mislocations of chlorine channel, which is called CFTR, which in turn dehydrates the mucus. It will also increase the viscosity of the mucus, and this is something that you could see here clearly, so that the beads are now moving, and this is clearly what you observe also in vivo in human, which are unfortunately suffering from cystic fibrosis. So you can recapitulate the aspect of this pathology in vitro. So interestingly enough, so how could we use that now? So in normal condition, if you have a xenobiotics or a pathogen, you could have a decrease of the cilia activity that you could be quantified here. Okay. Now, look at cystic fibrosis. Clearly, it could be used here as a platform to screen compound which could correct or activate the CFTR, restore the function here. So, which could be for efficacy assessment. Okay. Now, I will almost switch to deeper lung model. So we have the small air, which is also has a cilia also and mucociliary clearance transport. But if you look now, do micro dissections of the alveolar sacs, you can extract here pneumocyte and endothelial cell and co-culture them here in order to generate the alveolar model. But you could also isolate tissue resident alveolar macrophages, and I will discuss these models also in the later slides. So alveolar. This is a model that we launched recently, which is made of pneumocyte of type 1 and more importantly, a sustainable long-term presence of pneumocyte of type 2. This is one of the main problems in vitro, is to stabilize pneumocyte of type 2 because so they are type 1 of the progenitor of type 2, so meaning that in culture you ended up most generally when you see type 2, you will switch to type 1, and at the end you would have only type 1. But in this model, which took us 10 years of development, we achieved that to have, type 2, which could be preserved with long term. Clearly, this is the thing here with that could be observed with marker such as HT2 280 for to show the presence of CAT2, or podoplanine here for 481. So it's tight and could be used here in this test. As you could see, it was up to 50 days. Yeah. So okay, so what could we do with this model? So first we need to discuss about how could we apply compounds. So exposure system. So as I said, all of the models that I've presented are in transwell format, meaning that you could use different strategy in order to test your targeted compound. So first, we have a static exposure scenario where you could add on top of the epithelium liquids, solutions, or suspensions by pipetting directly on top of the epithelium if you are interested in apical exposure. But we have also developed an interesting way to apply to generate tablets also with a nanopress here, especially for highly reactive compounds. So this is a possibility to test also solids in a static exposure, but also dynamic exposure. So they are fitting with commercially available exposure scenarios. So we can test here gas smoke with, for example, vitrocell VC1. You can nebulize here with the vitrocell alpha. There is also compatible with the CIREC Expo Cube, and also within, for example, the new Poderix system. Okay, so these are the classical ways of exposure, but I have to say that most of the experiments are run directly with static exposure like this. Okay, so what could we do now? We have the tissues which could be shipped to India, ready to use. So we are talking about mucilaire, smaller, alveolar here, and we have developed a panel of in vitro assays, so covering, for example, nasal dryness syndrome, cystic fibrosis, mucolytic activity. They could be also used for asthma COPD. So we have developed assays for this. Anti-inflammatory compound evaluation, bacterial infections, pulmonary fibrosis, inhalation, drug delivery, viral infections. So I will not cover all this aspect, but some of them in the upcoming slides. So I will first concentrate on one of the most important, which is evaluation of in vitro of toxicity when you inhale something. Okay, so if you inhale a compound, you apply your compound on top of the epithelium. So as mentioned previously, you have the epithelium which is on top of this membrane. You have the pseudostratified epithelium. The epithelium is tight, and you have presence of the mucus on top. Okay, so you could apply here solutions or suspension directly. So if you want to mimic a systemic toxicity, you may apply your compound in the liquid. So basically, there is a wide variety of potential subcategory in inhalation toxicity that we have divided into local tolerance evaluations, mainly used for point of contact or point of departure toxicity, evaluation of barrier functions, carcinogenicity, disease induction. So we can name two diseases, for example, but there are more. So fibrosis, hyperplasia, for example, detection of inflammations, and sensitization. So I will cover some of them. But first, for the local tolerance, we use a panel of endpoints which are complementary. So we monitor here the impact on tissue integrity, monitoring transepithelial electrical resistance with electrodes. We can monitor here the cytotoxicity, quantifying what is the LDH which is released in the culture medium. We can also quantify here the inflammatory signature by quantifying the key inflammatory cytokines such as interleukin 8 and interleukin 6, and look at also the ciliopathic effect and evaluating the cilia beating frequency and mucociliary clearance that I introduced previously. So I will just show some data which are reported in the literature. So one of the first studies that I'd like to present is was published by AstraZeneca, and this is the title of the paper: 3D human model enable prediction of respiratory toxicity of inhaled drug so in vitro. So the idea was to compare in vivo and in vitro data and look at repeated dose exposure. So it's repeated dose for 12 days here, and look at a different endpoint. And interestingly enough, one of the most easy to monitor endpoint is here, transepithelial electrical resistance. And it appears in this paper, the studies, to be the most sensitive endpoint to predict toxicity. And in this paper, they reported that a very interesting 88% sensitivity of this model and 100% specificity. Also, there is still a moderate number of compounds which was tested, but 15 compounds. Still, this is really clearly encouraging. So now, another studies that I'd like to also to share with you is an achievement from a very long journey. Trying to here also link with data with the regulation, so this is something that took a lot of years here to achieve, but we were really proud to be to show that here the mucilary system has been used here and also has been endorsed by OECD as case study for replacement of 90 days rest study. Use here in order to test, to evaluate chlorothalonil, which is a pesticide, and especially to derive point of contact toxicity. So this has been the first replacement study which has been endorsed by OECD, and it was done in 2022. There is a second regulation which has been also published also, but now in Europe, by the SCCS, which is a scientific committee of consumer safety in Europe. And now the compound which has been tested here is acetyl vetiver oil, which is used in cosmetics as a fragrant ingredient, and mucilary data has been also used in this study. So now this model and data from this model are not only a fancy scientific tool, but they are becoming really decision-making tool here in this regard. Okay, I will now switch about respiratory absorption, because the airway epithelium is a barrier per se. This is his main activity, is to block pathogen when you breathe. But it's important to understand when you breathe something if it can cross the airway epithelium in a way, so this is something that we could do from all the parts of the respiratory tract, from the nose, trachea, bronchial, and small airways, and alveolus. And this is an example here, which has been here co-published with the European Center of Validations of Alternative Method, EGVAM, where 30 compound has been tested, 33, in different chemical classes. So it could be metals, you could find drugs, but also consumer goods. And the idea was to rank the permeability. Okay, so we ended up with permeability constant here, from the highest permeable to the lowest permeable compound. So how could we use this? So if my compound is highly permeable, that would say that it certainly could be considered in the to be present in the blood and circulated. So you had better here to test on other organs than like, for example, the brain, liver, heart, for example, models, because they might be found in the bloodstream. Interestingly enough, you can also derive from this data a dose that could be tested to minimize downstream doses to be tested later on in the in vitro assay. So you have moderate here permeability, and you have very low permeability. So how could we use this with very low permeability? Well, if my compound is not toxic, and if it's not permeable or quasi with very low permeability, the risk in terms of toxicity is really low, and this is used in this regards. Okay, so clearly this is an easy assay which could be could bring a lot of information. This is induction. So this is another topic. So this is induction. So well, one of those is metaplasia. So metaplasia. So is something which is clearly found. So in COPD, for example, which could it could be a hallmark of a long-term repeated exposure. So one of the marker could be used is MUC5AC area, so that MUC5AC staining. These are cross sections that you could see that we have this staining. So MUC5AC could be observed here in brownish here. And clearly you have the untreated the vehicle, but upon 2 weeks repeated treatment with IL-13, you ended up with this upregulation, like what you could see, observe here, and which is a block, for example, with a positive control, an antibody, lebrikizumab, for example. So as a consequence, also the ciliation is also there is less cilia activity, and you could see that cilia beating frequency is also quite perturbed in presence of IL-13 in this way, which is also prevented in presence of the lebrikizumab. So this is an interesting model also to look at this potential harmful effect. So we have also developed a 3D confocal analysis that I will not go into the details now, but we can also monitor now on 3D, not only on 2D. Okay, so I will now jump to other type of applications, which are pulmonary fibrosis and also infection models, because it has been used quite a while. And we'll start with viral infections, and this in vitro models, so mucilaire, smolaire, and alveolar, has been extensively used to test the effects, pathogenicity of different respiratory viruses, such as rhinoviruses, for example, family, influenza, respiratory enteroviruses, coronaviruses, and especially SARS-CoV-2, as you may imagine, during the pandemic, but also respiratory syncytial viruses, and some other important metapneumoviruses, parainfluenza viruses, and there are also some program on Nipah viruses as of today. So the idea is to test what could block, to some extent, the pathway of replications of these viruses, and to develop, obviously, novel antiviral strategies. So, but clearly, the process of these respiratory viruses is very simple. First, the respiratory virus will just link and be kept through a cell surface receptor present on the apical surface of the membrane, which will lead to a virus attachment, and then incorporations of the virus, which will release is viral RNA, which through reverse transcriptions will here integrate here. Then we have integrations and again transcriptions, and then translations, and then assembly to assemble a new virus core, which could be spelled out and released on the apical lumen in the mucus. Okay, and for this reason, when you this from one, you can generate many different viruses inside the cell. So this leads to a system which is exponential, okay, in terms of in terms of replications. So by the way, this is clearly important if to have a non-transformed cells here to have something which could bear the right receptor. And for this reason, at Epithelix, we are working with only low passage cells for all the models that we have to ensure that we have efficient replication. So I was mentioning exponential growth, and clearly this is things that you could see. So here I represented some replications with time, for example, over 1 week. So this is in hours, so 20, so until 1 week here, and you could see the replication of some viruses such as influenza, H1N1, H3N2, some enteroviruses, and also rhinoviruses A16. You could see clearly a very nice dose, sorry, replication, which is time dependent for some viruses. And for each of these viruses, you can inhibit the virus growth using a specific antivirals. For example, a 3C protease inhibitor in case of Ripenctivir. You could see also Zentamivir and others here to prevent replication of the influenza viruses. So there have been extensive also publication on COVID, but I just want to mention one of those, which was the ranking of combinations. So we are happy also that to see that some you could find also some antiviral which has been like which had been used during this break. So remdesivir is molnupiravir, so has been here shown, and Paxlovid here. This is what you could see here. Have been evaluated successfully in this model, and you could also test some B or tree therapies. There's some papers around that. Okay, so I will now switch to from viruses to bacteria, and more specifically look at antibiotic efficacy, evaluating using mucilaire, smolaire, and alveolar. So these are the different in vitro infection model, such as Pseudomonas aeruginosa, Streptococcus pneumoniae, Staphylococcus aureus, Haemophilus influenza, Bordetella pertussis, Acinetobacter baumannii, as example. So I will focus on one of the most devastating, which is pneumoniae. It came from a collaborative project for European collaborative project from which is called FAIR, for flagellin aerosol therapy. So the idea in this model, so is first we infected with SP90F, for example, which is a Streptococcus pneumoniae strain here, and we added flagellin therapy, which is TLR5 activator here, inducer of the innate immune response of the airway epithelium. And the idea here is to boost the activity of some antibiotics, because amoxicillin here is clearly not working enough, especially on resistant strain. So the idea is to boost the innate immune response. So what you could have shown here is that we have obtained a modest inhibition of this bacterial growth, but some defense mechanisms such as beta defensin 2 and IL-8 have been shown in the model. And clearly, really interestingly, and we ended up because we had also a clinical trial based on this, and this marker has also been reported in human to be upregulated in the clinical trial. So we are all in the consortium very excited about the data generated in vitro to see the translation. Okay. I will now switch to alveolar macrophages. So we isolate alveolar macrophages in the alveolar sacs. So they are resident, tissue resident alveolar macrophages. Which are cryopreserved, and we ship them and they are ready to use here, and they are suitable for airway co-culture models. So they have shown here to be also they have a phagocytic activity which could be observed using, for example, a pH Rodot zymosan test. But not only so we have run also some study with this. Look at also the phagocytic aspect with fibers. For example, this is an example with coccidolites, where you could see that they undergo frustrated phagocytosis on top of the alveolar model. But interestingly, they could also break some fibers, such as the glass wool, for example, which you could see the breakage all along the fibers using these alveolar macrophages. And these data are consistent also with observation of in vivo. So how could we use this also? And there are quite interestingly so this is an example again with the SP19F that I mentioned, the pneumonia strain that I mentioned previously on the mucilary system. Okay, and now if you have the macrophages, clearly the macrophages are reducing really largely the growth, inhibiting the growth of the bacteria, and interestingly enough, they are also preventing some secondary endpoints and signals, such as the mucin secretions, for example, which is upregulated in presence of SP19F, but not in presence of the macrophages. So if you look at here, so this is the type of inhibition that you could see in pneumonia infection. But if you look at another bacteria, such as so here Staphylococcus aureus infection, so the inhibition is quite less important. So suggesting that the inhibition is bug specific or bacterial specific. So this is something that it's still under investigations and in terms of and understanding. I will switch now to idiopathic pulmonary fibrosis. So, which is also devastating. So the idea, so this is mainly driven, in the alveolar sacs, and there are two main functions that we want to recapitulate: is the epithelium to mesenchymal transition due to the challenge, but also the fibroblast to myofibroblast transitions, which here induce excess of collagens and really real problem of our breathing later on. So how do we do? We use 3 days models here in order to test antifibrotic drugs. So we fasten the process by adding a profibrotic cocktail, which is a mixture of TGF beta and TNF alpha, and co-culture here with the antifibrotic treatment. And we see it here also with primary fibroblasts that we also could provide. So the idea here is that at the end of the 3 days is to monitor a panel of endpoints which are clinically relevant, and I'd like to show two of them. So the first is epithelial to mesenchymal transition. So the fact that you change morphology here. Of the epithelium as found here, and adding stress fiber, actin stress fiber. And one of the marker in clinics are MMP3. So this is an example. So you upregulate MMP3. So we are talking about secretion of MMP3. And clearly you could see that, for example, anti-fibrotic drugs could, in a dose-dependent manner, reduce the nintedanib, and clearly also it acts also on the MMP1. So another type of marker are the FMT, fibroblast to myofibroblast transitions, which consequently lead to excess of extracellular matrix synthesis. So the way we do is to quantify here alpha smooth molecular actin here, so that we could quantify by fluorescent microscopy. And we could also look at, for example, some gene of expression. So this is the way we run. So just to sum up now, so all in one, what we could propose at Epithelix is so as I said, nasal, tracheal, small airway, alveolar models. So the model which we call mucilaire, small air, alveolar, they could be derived from normal COPD, asthma, cystic fibrosis, or allergic donor if needed, from single or from pool of donor if needed. We can also have some co-culture with fibroblasts, endothelial cells for some models. But you could add also immunocompetent compartments such as the macrophages or the neutrophils that are not covered today. But it could be also co-culture with either dendritic cells and PBMC in the lower compartment with a serum-free medium. So there are a lot of different possibilities, and as a consequence, it's fair to say that nasal, bronchial, small airway, and alveolar 3D human models are useful tools to evaluate effect of therapy on local tolerance, respiratory absorptions, mucociliary clearance, mucosal inflammations, detections of onset of chemical-induced respiratory diseases such as COPD, pulmonary fibroblasts, for example. And still, assay needs to be developed an emergent immunocompetent model integrating alveolar macrophages, dendritic cells, and neutrophils. So I'd like to thank you all for your attentions, and thank also all the team from Epithelix, from our French labs and also our Swiss labs. I would like to mention especially two colleagues, Charlene and Shoyan, who are present today, which could help you, for any question also you may have in terms of logistics. They're key element at Epithelix to help our customer, and not only. So I would like to warmly thank them. And I also invite you all to a congress that we are organizing, which is called the Lung in Vitro event, the Live 2026, which is close to Geneva, which will be the 8th and the 9th of October. It's a 2-day conference where we only work on discuss lung in vitro and from discovery to prediction, and there are some opportunity to present poster, to have talks also. It's a 2-day, and there is also linked with hands-on training. So that's all I wanted to say. Again, thank you for your patience, and I'm open to any question that you may have. Thank you.
Thank you, Dr. Samuel, for your very nice presentation. So we have some questions from chat box and the seat questions that we have prepared. So first question is, how do ALI-based primary human airway models compare with traditional submerged cell cultures in predicting clinical respiratory toxicity and drug response? So this is the question from audience.
This is a very interesting question and very important. So first, when you think about inhalation toxicity and relevance, it's important to have the right type of cells first, and also the right phenotype, meaning that the tissues or the cells should have mucus, because the first thing that when you inhale something, it will touch the mucus. If your model has no mucus, then there is a strong irrelevance. There is also the cilia activity. If your model have no cilia or less cilia, it will not recapitulate some aspects of the inhalation process. So these are key elements that needs to be have, it's a have to have. It's not a nice to have in the model. So that's why the mucillar, for example, for the upper respiratory tract, is widely known for its here quality in recapitulating this aspect. And this was, as I presented, this is the first model which has achieved regulations through the chlorothalonil clase with OECD worldwide.
Yeah. So this is the question from most of our users we used to get from every day. What are the key challenges in translating data from in vitro ALI airway models, nasal to alveolar, into regulatory accepted endpoints for drug discovery or development? Like, what are the key challenges you face?
Again, this is a very interesting question, because so obviously the main challenge, for any in vitro model is the fact that it should be transferable. The model should be transferable. You should ensure the quality. You should have stringent quality control, and this is something that we have first for transfer looked at heavily. So, for example, all of our tissues we monitor. We look at if they are this epithelium is tight, and when our customer can ensure that using simple measurement with transepileptic electrode, if the tissues are in good shape. You could visualize if there are cilia, because the insert is transparent, and you could see by simple eyes if you have cilia motion or not. You could also look at the presence of the mucus by refrigerancy index. So these are quality step also, which could ensure the good maintenance of the epithelium. And also, you mentioned regulation. So there needs to be let's say produced into a framework which is also acceptable for the regulations. For this reason, all epithelics products are produced in good in vitro methods practice. So GIVIMP, we are accredited by GIVIMP to ensure this quality. Now, once you have all this in place, it's question of generating an assay. And the assay is also, it takes a lot of time in order to look at data and compare in vivo to in vitro. I've shown you case study for AstraZeneca. They have compared in vivo, in vitro. They ended up with a very good sensitivity and specificity. And you need to compare, ideally not with animal, but ideally with clinical data, epidemiological data, if possible.
Yeah, very nice. So last question is, with now increasing emphasis on a new approach methodologies like NAMS, how do you ALI airway models contribute to reducing animal testing while meeting global regulatory expectations? So how you see yourself?
Uh, Well, this is a mission of Epithelix. We created Epithelix in 2006 with the idea because we saw that animal models were not enough predictive for the health and also the efficacy of human. So this was clearly something that we could observe. We are not the only one. Many are also under this agreement. But at this time, this is what we could do. But now we are in 2026, and we have done so many better models so that could be used here and are used. And I've shown you different example. So for tox, when you inhale something, nasal spray, or anything, occupational, the first point of contact toxicity is on the airway mucosa, and you have here the possibility to have toxicity on different region of the respiratory tract. It's not a black box like an animal. It's much easy. It's less expensive, easier to control, and you have human information. So these are cases where clearly there are much more advantages of using this. So we are working hard at Epithelix, and not only us, all the users of our models worldwide. So as I said, so our models are heavily used in Europe, they are heavily used in North America, they are heavily used in Asia, Japan, South Korea, for example. And we are working on case studies, like, for example, I've shown fibrosis, and we publish, and then we go to the regulations, and in turn, at the end, case by case, we will reduce the number of animals to be tested. But we are in a very good momentum because we have a driving force from the politicians, from the FDA in particular, from the Europe also, and I think that worldwide, it's no more a question of recognizing that these NAM are useful. They are useful. It's more question of putting boxes and very clear assays for each of the topics that I mentioned.
Thank you, Dr. Samuel. It was a very nice and insightful presentation. I also want to thank all the attendees for attending this webinar today. We are also grateful for the participation and hopeful that you will find this session informative and engaging. We appreciate you taking out the time from a busy schedule today. Your engagement and thoughtful questions contributed greatly to the success of this webinar. And we have a special thank to Dr. Samuel and team for sharing their valuable expertise and insights on advanced airway models, their human relevant design and application in toxicity assessment, drug safety evaluation, and respiratory disease modeling. Thank you so much. Thank you. Bye, Dr.
Thank
Thank
you.
you.
Thank you
Yeah.
very much.
Yeah.
Bye.
Bye.
Have
Bye-bye.
a nice day.
Bye.
Bye-bye.